Welcome everyone. I will talk today about tools, workflows, and infrastructure for open, reproducible analysis of SARS-CoV-2 data. We will cover genomics, evolution, cheminformatics, and a few other projects that have been developed by the global Galaxy community since the beginning of February. Galaxy is an open source framework for reproducible data-intensive research. It's more than 15 years old, and it's concentrating on delivering, accessing, and sharing data, tools, and workflows. Galaxy has multiple user interfaces. The most prominently one is probably the web portal, so a website where every researcher can upload data, start workflows, start tools on top of it, visualizing the results, and then finally sharing it with the community. But Galaxy also offers an API for bioinformaticians or users that want to use Galaxy features from a command line. Galaxy also provides access to HPC and cloud resources that were fundamentally for the work we will talk here about. Moreover, Galaxy is an international project, so we are at least aware of 130 public Galaxy instances, and all of them can be used freely for everyone that want to start data analysis. We will cover today a few topics, and because of limited time span, if you want to know more about all these projects, if you want to have the results, if you want to download the results, if you want to start workflows, in general, if you want to have more detailed information, please visit the covid19.galaxyproject.org website, where we have collected a bunch more information. So let's start with the genomic aspect of SARS-CoV-2. So since early February, we started to build workflows and started to process the publicly available datasets. So for example, we spent a considerable amount of time in pre-processing workflows and pre-processing the publicly available datasets. Unfortunately, you will find a lot of datasets that have bad quality and you should not really use them in your further downstream processing. But for example, you will also find annotated SARS-CoV-2 datasets that don't contain any read of SARS-CoV-2. So all these pre-processing and quality control we have separated in one workflow, and you can just use them to clean your data and to get an appropriate quality report out of it. If I'm talking about cleaning data, we have especially put some effort in creating a cleaning data set that will separate human and SARS-CoV-2 reads so that you can be confident that you don't upload human reads to public archives, for example. We also created an assembly workflow that can create a consensus genome, for example, but also workflow for most recent common ancestor analysis. We have created workflows for variant analysis of the virus, which we use to process actually all publicly available data sets that are available. So we are talking here about more than 20,000 complete genomes that are available in public archives, and we use these variant analysis workflows to crunch regularly on these data sets. We have a data set for the S protein analysis, which will also create nice visualizations for you, so you can really dig into the differences between different data sets and find differences in a visual way. And we have an entire subsection about the evolutionary analysis, outcomes, conclusions, and you can browse them interactively in a notebook. So all these workflows are open source, are building on community projects, are installed via Bioconda and Biocontainers, and you can use them in any of the big Galaxy instances, which I will cover later on. One challenge is that data is released very frequently, which is super fortunate for us because we can more or less, for the first time, track the virus in real time. This is also a challenge for computational research and to keep up to date. So what we have done is we created a small bot that is sitting on GitHub that is reading and filtering metadata from public archives and updates simple flat files on GitHub with accession numbers that belongs to SARS-CoV-2. Given such a pull request from the bot is merged, the automatic or the continuous integration setup that was developed will automatically kickstart workflows in different Galaxy instances so that the other day when you wake up, for example, you will get a new result of very, very recent data that was published maybe a day ago. One of these green columns that you are seeing here is a so-called galaxy history. And a galaxy history contains all provenance information that you need for a galaxy workflow. For example, the tool that was used, the tool version, every single parameter setting, and finally all inputs and output files. So we will provide you here in an automatic way, based on our workflows, the very recent VCF files from the variant annotation workflow. You can also download the BAM files as you like, or every intermediate step. This entire setup not only works for whole genome sequences, but for example, if you have amplicon data, we have a specific amplicon workflow, for example, for the Arctic protocol, that you can treat in the same way than the variant workflow. Let's talk about cheminformatics. This is another project that we have been developing since a few months. And it deals with in silico drug design. And it's part of the International Moonshot project to find a drug against COVID-19. At first, we needed to identify a druggable target. And one of the druggable targets that we can target is the main protease. In cooperation with the Diamond and with the synchrotron in the UK, we had access to a crystal structure of the main protease. And Diamond approached us to develop a workflow that is reproducible and that is open from day one to actually do a fragment-based drug screening. So we had some fragments that were crystallized in the MPro crystal structure. And from these fragments on, we enumerated the compounds, we prepared the ligands, we enriched them, we generated 3D structures, we created or prepared the proteins, and finally we did some docking experiments that we later on, the poses we used in some scoring and ranking model to finally come up with top 500 of compounds that we are now trying to synthesize and then finally go into in vitro testing. This effort is very huge in numbers. For example, we had nearly 160,000 candidates and 17 proteins. So the Diamond is producing more proteins and more candidate structures as we speak. So we also need to rerun these workflows more or less constantly on new data. But essentially for 17 proteins already, we have nearly 3 million docking jobs that we executed, and nearly 70 million of different poses that we generated and that then needed to be scored with different ways to more or less filter out or have the 500 most promising candidates. The fun fact is here, and this is also demonstrating that the system and the framework is really capable also to use GPUs, is that one of these scoring functions can only run on GPUs. So we have here access to more or less a hybrid cluster where we submit jobs either on GPU nodes or CPU nodes, depending on the input data sets or the tool that is running. We have a few other projects that I would like to introduce you very briefly, but again, due to the limited time, I really encourage you to get in contact with us or visit our website. One of the projects I would like to mention is a collaboration with the EMBL and EBI. And especially with the Human Cell Atlas, that created workflows, tools, and visualizations inside of Galaxy, and especially also to process COVID-19 datasets. So if you have single-cell RNA-seq datasets that you want to process and that you want to compare, for example, against the Sanger COVID-19 datasets, you can use one of these workflows and visualizations to compare your data against one of these public resources. Since there is already some open access proteomic datasets available, for example in Pride, the Galaxy proteomics community created workflows to reanalyze these workflows in a reproducible way and put on top some post-processing steps, some visualizations, and they enhanced the analyzers already in respect to doing a meta-proteomics analysis. Another project which we are currently working on is the direct RNA sequencing workflow. So there are only a handful of samples currently available worldwide, but I think understanding the epigenetics of the virus, and especially, for example, RNA modifications, is at least scientifically interesting. So we created 2 workflows. One is based on NanoCompor, and the other is based on Tombow. And you can run them on the few publicly available datasets, which we also mirrored on our Galaxy instance, so that you don't need to download a terabyte of data. It's all stored there, and you can crunch these datasets and try to understand the epigenetics of the virus. All these workflows and all these tools are available for everyone. But Galaxy is providing more than just workflows. Galaxy is also providing the infrastructure to execute these workflows, and this for everyone. And this brings new challenges. So for example, one Galaxy server probably doesn't scale to millions of researchers that want to analyze hundreds or even thousands of data sets. So what we are doing, and also to utilize all our local infrastructures, compute infrastructures, is that these workflows which we have talked here about are deployed in several Galaxy instances worldwide. So we have usegalaxy.org, and the workflows there are also available in usegalaxy.org EU, so that's the Australian Galaxy server, or the European Galaxy server, usegalaxy EU. The workflows are the same, are deployed in different Galaxy instances. These different Galaxy instances can use different compute resources, but the results are still the same, are reproducible, and you can choose wherever you have your resources available or which Galaxy instance is the nearest to you. Another approach which we have developed since a few years, but that we extended during the pandemic extensively, is the Pulsar network. The Pulsar network is kind of a distributed compute environment for Galaxy. So for example, the European Galaxy instance is able to submit jobs to the UK, to Finland, even to Australia. A user that starts a variant calling workflow on the European Galaxy server can distribute the jobs to other HPC and cloud centers in Europe. Galaxy is managing the compute infrastructure for that, and the user can really concentrate on the research question. So the user doesn't need to care about the compute or where the compute actually is happening. If you want to learn more about the webinars, the workflows that we have presented here, we have created a bunch of webinars with the Galaxy project and the Elixir project together. So each of these webinars is 1 hour, and if you want to learn more, you can also just watch one of these webinars, or, as I already said, please get in contact with us. Last but not least, I would like to point you to some training material. So we participated in already a few hackathons where people wanted to learn more about these workflows and how they can analyze data sets, SARS-CoV-2 data sets. So we created over the last months, a few dedicated tutorials, e-learning materials, specifically for SARS-CoV-2. So if you want to learn how to assemble your genome, if you want to learn how to do a fragment-based drug design screening, check out our training material. You will get the e-learning materials, so hands-on material. You will get example data that is downsampled, so you can actually process that in a few hours. You will get, of course, the workflows, and you will get the compute resources to do your training. With that, I would like to thank my team here in Freiburg, but the overall Galaxy community as well. So the Galaxy community is fundamental to the success of these projects, and also that we could deliver all these workflows in such a short amount of time. So most of the tools, most of the infrastructure was already in place